Well, maybe while we're waiting for Asaf to join, I'll start by welcoming everyone to March. It sort of came in not like a lion or a lamb, I would say, somewhere in the middle. A lot of things happening. We'll hear from our speaker in a couple of minutes. We have a neuroprosthesis seminar coming up in April. Ray Silito from Baylor. We might have one in March if a certain someone actually does what he is supposed to do. See, next week on Tuesday morning and Wednesday morning, we will be having a virtual site visit for the FES Center by the VA Rehabilitation Research and Development Service. This is associated with the renewal of the FES Center. And I hope this is the final step. Just to sort of catch people up, we submitted a proposal February 2021. It was reviewed quite favorably. We had a score of 130. That's on the old NIH scoring scale, which is really good. We've responded to the reviewers a couple times. There were delays about getting a site visit scheduled, I think because of COVID and interference with holidays and availability of reviewers, etc. So this is finally happening. If you need to be involved, you know about it already. Again, it's virtual, so it's a little bit unusual. Yeah, if you're interested in attending any, well, not any of the sessions, some of them are closed, but we have some sessions that have to do with presentation. I get 15 minutes to present the center, so I wouldn't get your hopes up about a lot of detail there. But we have other sessions where young investigators are going to be meeting with them. They're going to be meeting with the leadership of the medical center, etc. So if I need something from you, you'll be hearing from me. Otherwise, think good thoughts on Tuesday and Wednesday next week. And hopefully we'll be announcing soon that we're funded for another 5 years. Okay, so that's the FES Center update. Asaf.
Yes, hi.
Are you in a position to introduce Robert?
Yes, I am.
Okay, take it away.
Thank you, Dr. Kirsch. Welcome, Robert. Welcome to the big family of FES Center. I want to take this opportunity to invite Robert Adams, who was a STAR resident and now a STAR fellow in our Department of Neurology, and very soon he will be a STAR investigator of FES Center. Without further delay, Robert is going to speak about neuromuscular ultrasound techniques for estimating muscle contractions in FES experiments. Robert did PhD from St. Louis, and his focus had been somewhat very relevant to FES. And then that's how the whole conversation and interest sparked. So Robert, all yours.
Thank you. So I'm going to talk on a topic today of an idea for research coming up. We don't have much in the way of preliminary data, so it's kind of to discuss ideas that we have going forward. Dr. Kirsch and the FES and rehab team, Baloo, Emily Graysick, Bill Memberg, kind of brought me into the rehab FES project that I've been working on, and we've been generating new ideas kind of for how to add more to that project. So for the, and then I think there are just a couple people overlapping with neurology. I gave pretty much a similar presentation to Grand Rounds like 3 or 4 days ago. So for those that were there, sorry for the repeat, but I don't think there's too much overlap. And so for the rehab project, that's a lot bigger than me, and a lot of people have been working on from multiple aspects of recording the brainwaves to interpreting those brainwaves, and then stimulating peripheral nerves the part that I've been helping with and working on and getting interested in is the how do we measure the muscle activity whenever we stimulate peripheral nerves in this rehab project. The way to get information for stimulating nerves to look at the muscle activity to hopefully kind of get control over a hand and a peripheral nerve stimulation. First starts with the nerve cuff that's around our peripheral nerve, and that stimulates the axons. Midway down the axon, sends an action potential in both directions, but what we're interested in for these functional electrical stimulation is the axon that goes to the muscles. That stimulates a few muscle fibers in one specific muscle for each axon. And then with the right combination of muscles stimulated, the idea is to get functional movements of the hand. So the challenging part of this algorithm is that we know the hand movements that we would want, and we have to figure out, what electrodes to stimulate for how long, at what intensity, at what paradigm, at what pattern to get those actual desired functional movements. Typically, to create an algorithm like that, the modern method is neural networks or other kind of deep learning networks. And to generate those algorithms, it just needs a bunch of data in the opposite direction of, so a bunch of simulations of a bunch of contacts, and then seeing what happens with motion, and then letting the neural network work backwards from there. I think the way that we're not doing this, because I don't think it's the right way, is looking at hand movements alone, because hand movements after electrical stimulation are based on where the hand currently is and what loads are on the hand. So creating a neural network or deep learning to try to get a hand movement just based on electrical stimulation, I don't think is the right approach. Isn't what we're doing. So we're kind of breaking that down into two algorithms. One is electrical stimulation to get muscle activation. And then the second step would be based on where the hand currently is, or based on the loads that are currently on the hand, what muscles do you want to activate? So the part that I've been working on or helping with the team on is just that kind of part of the whole algorithm of when we stimulate electrodes, what muscles are activated, and to be able to then eventually generate that neural net algorithm of desired muscle activations to electrostimulation paradigm, we have to actually have a good way of recording muscle activations. The classic technique for this, and the technique that we're using right now regularly, is with electromyography, EMG. That's done with a needle that's inserted into a muscle. And then when a motor unit fires, that axon is hooked up to several tens to thousands, depending on the muscles, typically in the hundreds range, of individual motor fibers, which are the most muscle cells. And then the combination of those motor fibers that fire in the proximity of the needle, kind of based on how close they are, create that EMG signal. And so that EMG signal is somewhat proportional to the number and size of the muscle fibers that fire. It's not a perfect analogy, but bigger EMG signals correspond to more motor units firing, or more muscle fibers firing, and smaller motor units correspond to smaller amounts and sizes of muscle fibers firing. So EMG has classically been used to kind of develop these recruitment curves of stimulation to how much muscle activity we're getting, and that's the method that's being used right now. The drawbacks, though, to using EMG to acquire this data are primarily just how many muscles are in the forearm. So here's a list of just forearm muscles, and that doesn't even include, if we were interested in the flexor digitorum sublimis and profundus, going to each of the fingers individually, because that'll break down those muscles into 4 separate muscles, or the extensor digitorum communis breaking into the 4. So that would be even 9 more muscles if we want to record muscle activity to all of those muscles. Other problems with EMG is typically when it's done clinically, unless it's just a big obvious muscle like a biceps, we use a little bit of assistance from the patient to identify the muscle we're going into. So if we know we're going into flexor pollicis longus in the mid forearm, we'll have them gently flex their thumb while we're inserting the needle so we can hear that electrical activity, and it'll sound like distant gunfire whenever we're outside the muscle. And then as we get closer, it'll be kind of the close reports of gunfire being sharp, and then we know we're in the muscle. So it's tough in an SCI patient in the rehab project because they don't volitionally produce any muscle activity. In this case, in these experiments that we're doing, we used ultrasound to identify the largest part of the belly of each of the muscles, and then the depth of each of the muscles to then give us some visual feedback for getting to the muscle. But unfortunately, you know, it's still a drawback to using EMG is that during each experiment, we don't know if some muscle belly shifted or something beforehand. So these are just some of the drawbacks to doing this technique for getting all those fine muscles of the forearm. And then not to mention the hand, which has another 19 muscles in it, which would be really difficult to EMG. So alternatives to EMG. One would be to just look at motion of the hands. The easy thing is probably just that this would be relatively simple to set up. The drawback is that it kind of skips 2 steps in that algorithm where motion itself can be tough. If the patient's hand is already flexed and pronated and you stimulate a muscle, it'd be hard to tell what action is going on. So it's based on the current position of the hand, which is why it's probably not the best technique. Dynamometers or force measurement is probably a good technique if you could create some sort of device that can measure the torque at every joint in the hand. And I haven't had that, and I don't think that would be easy to do, but it would probably work. Force is a good estimate of muscle activity. And then the method I'm going to be talking about here is ultrasound, which may be a relatively quick and reliable setup that can be moved between patients. Ultrasound has really come to fruition. I think maybe you'll find this talk interesting just to kind of learn a little bit about the techniques that we're using now with ultrasound. It's come to be mostly in the past decade. The technology's improved enough at this point that we can actually see the individual fascicles of the nerves and the muscles quite well. So we use it clinically. I probably do about 40 studies a week diagnosing different nerve and muscle disorders. Then physiologically, for research applications, that's come to be over the past 10 to 20 years as well. Not used too often in a variety of kind of physiology labs that are trying to look at muscle activation during gait and this and that. They use ultrasound to estimate muscle activation. That's where almost all of the data will be. Also, occasionally abdominal muscle activation, occasionally lumbar muscle activation. There's a few labs that have been using this for the past decade. Most recently, or the past couple decades, most recently, there have been improvements in automated, deep learning algorithms, as well as a new technique called supersonic shear wave elastography, which is, I'll just kind of prove to you a little bit that we are able to look at all the muscles of the forearm and that by applying an ultrasound, we can pretty quickly determine where all the muscles are. So if we look at the distal forearm, we can see the flexor digitorum sublimis and profundus and the flexor pollicis longus. And if we go up a little higher, we see the pronator teres and flexor carpi radialis. Or if we look at the back of the arm, we can see all the extensors as well, just from one plane. So you can get, all in all, you can get all of the flexors and extensors of the arm and forearm in 3 spots.
Give us some idea of how long it takes to get that. Is this just a flash picture, or you have to? Because every time I've been around ultrasound, they're moving it around all the time. And what does it take to get this picture?
A flash picture. So putting jelly on the probe and then dropping it on the arm, and then it would be there. This is coming in at about 50 hertz. So immediately, no post-processing required. But all the colors here are post-processed. That's me putting that in. This is what it looks like immediately on the fly. And then one thing we can do for a more complex case or or muscles that seem to kind of merge together that are hard to tell apart is you can have people flex certain muscles in their forearm, for example. So that's me flexing my thumb, index finger distally, and then proximally, index finger distally, and then proximally up here. Or middle finger, I'm sorry, ring finger distally, and then proximally up here. And then little finger distally, and then proximally up here. So even though FDP and FDS are commonly considered one muscle, we know the different fascicles obviously go to each of the fingers, and those can be distinguished based on ultrasound. And then I'll just let you watch that in the extensor department as well. So that can be done actively, but it can also be done passively quite well, where just flexing the finger, extending the finger can cause movements in the muscle. And even just kind of absorbing that video for a moment, the idea that intuitively we can tell kind of which part of the muscle is moving, that will lead to some of the algorithms, the ideas that we can use that intuitive idea to then get quantitative ideas of how much the muscle is moving after activation of the nerve nodes. To understand the parameters that others have looked at for the muscles, it's just important to know kind of what a muscle is made of. The main point of this is, they have the sarcomere inside each muscle fiber. When the sarcomere contracts, there's conservation of volume. So every time a muscle shortens, it will get wider as well. Whenever we're ultrasounding, we look at echogenicity. That's a common thing clinically. I don't think it'll be too important in these FES experiments, but just so you have an idea, when things are dark, it usually just means there's water and fat or something kind of homogeneous, or if something's extra bright, and there's extra collagen or fibrous tissue. And then this is all one muscle. One of my patients, brachioradialis. The white here is fibrosis due to myopathy, and then the dark is what normal muscle looks like. And then we look at the echotexture as well. That's just a fundamental property that others use in their physiology experiments to quantify muscle activity. If you look at a muscle in cross-section, a little bit higher up on the arm here, that's the flexor carpi radialis, pronator teres, flexor digitorum sublimis, and flexor digitorum profundus on here. And so what you can do is rotate the probe 90 degrees. So it's long axis. So you can kind of see if you put rotate it 90 degrees on this orange bar here, you can see the lines in between those three muscles. The echotexture, those lines are essentially the collagen fibers that run through the muscle. And so it looks like a starry night in cross section, but then if you run it longitudinally, you get those longitudinal collagen or fibrous bands. And then the shapes of the muscles will affect what properties we can gather as well. So for the most part, muscles are either pennate or parallel or fusiform, and the pennate essentially gets more force and less distance for its size, and the fusiform gets a little more distance and less force for its size when it contracts. And it just shifts differently, as you can imagine, whenever those fibers contract. So these are essentially up to date. So up to now, these are all the methods that people are using to quantify something that's observed on ultrasound to this is how much EMG activity we have, and we can use it as a substitute. One of the reasons that people often put in their papers is they have trouble getting IRB approval to stick needles in people for the experiments they want to do. So often they're looking for some less invasive form to get a little bit deeper muscles, a form of measuring muscle activity. It essentially falls into either muscle movement categories or muscle force, which is more new and less well established. So muscle movement doesn't fundamentally mean EMG activity by any means. So if you could perfectly fix a muscle and you grab both the ends of the tendon perfectly fixed and then you flex that muscle, it technically shouldn't move because the muscle itself isn't moving at all if you have it perfectly fixed. So all these aspects of muscle movement, the pennate angle and everything wouldn't move in a perfectly fixed joint. However, all of the experiments you'll see that it does, and that I think if we then think one step further, think physiologically when you start to flex a muscle, first you pull slack out of the muscle and joint and tendon. Next, you start to stretch the muscle and joint as the sarcomere continues to contract. And then eventually they found in most of these experiments, beyond about 20 to 30% EMG activity, you don't see much more of these kind of movement parameters changing. You don't see it shortening, you don't see it widening, you don't see the angles changing. Whereas techniques that look at force will continue to go up as the EMG activity goes up. Essentially every aspect you can imagine of a muscle contracting, so whether it's shortening or widening, or using Doppler to look at the speed at which it's widening, or looking at the angles of these lines whenever it's a pennate muscle, can be used to estimate that movement of the muscle. Another newer technique, which I thought was clever and will somewhat apply to a method I'm suggesting, is a lab used Doppler, and they angled the beam, which you can do using some math on the piezoelectric probes on the Doppler. They angle every other beam to slightly forward and the next beam to slightly backwards. they calculated Doppler on each of those beams, and then using some vector math, they can figure out the like translational velocity, which is the opposite direction of a standard Doppler for ultrasound. So just kind of for Doppler on ultrasound, it's always either how fast something is moving towards or away from the probe. So people are coming up with more clever techniques to figure out Doppler along the axis of the probe, which is more applicable to most muscle movements. And then lastly mentioned, just increases in cross-sectional area as a common technique. A new technique, which isn't standard on all machines yet, but is starting to be, is called shear wave elastography. The way that it works is they use the ultrasound to create a push. And so they activate all the piezoelectrics at the right sequence of times to create a converging ultrasound beam that actually like hits one point really hard, and that's the push point. And then that push moves along tight collagen fiber in the length axis of the muscle. And then as that push then reverberates, it creates a shear wave that bounces down that tight collagen fiber. It echoes back, and that shear wave moves faster than the typical speed of sound. So then as it comes back, they can figure out how tight that fiber was. It's actually a pretty reliable technique. Once it's calibrated to muscle, it can tell you how much force is in the muscle, and that can be calibrated to the electrical activity of a muscle reliably. And here's an example of measuring the force on somebody's gastrocnemius as they're pushing on a push pedal, and the tension they're measuring in the muscle is very closely aligned with the actual torque in their muscle compared to the pedal. A similar technique, but more applicable to what we would want to do, though not fully established yet, is doing this in cross-section. So in this case, I mentioned that the muscle is longitudinal, where they create a push, it moves down the length of the muscle, and it gets reported back. What we want is probably cross-section, so we can see all the muscles at the same time. So a lab created pushes in the muscle, but then let a deep learning algorithm just see all of the echo waves that came back and then had it calibrate itself. And then this is an example of images they're getting of the gastrocnemius on top here and the soleus underneath as a patient or as a research subject was doing a squat and while standing on their toes. And so whenever your legs are extended, when you're standing on your toes upright, your gastroc is most flexed, and you can see that is the most flexed up here. And then as they get deeper into the squat, the soleus becomes more flexed. And then the opposite happens as they come out of a squat. So
am I to understand that this is with deep learning? This is fast enough that you can get this where it looks real time?
Yeah. Oh, interesting question. I don't know if that's real time. They can acquire the data at over 50 hertz and then put it into a deep learning algorithm that then gives these images that at the experiment at 50 hertz, they have these images. But I don't know if it's coming up colored on their screen at the same time. I think it needs to be post-processed. And then a last kind of technique, not even a technique, but just what another lab is doing using deep learning. They were doing longitudinal images just of a muscle itself. And this is from their supplemental data saying that where they had patients hooked up to a pedal that was moving on its own, and then they had an EMG hooked up to their calf. So they had to match their EMG activity with the goal EMG activity while their foot was passively being flexed. And they said, look at this gastrocnemius moving. Can you tell when it's flexing, and can you tell when it's passively moving on its own? And part of the point is not necessarily, but there does seem to be kind of two different types of movement in there. Some may be passive and some may be active. So they just used each raw data, not even giving it sequentially to the algorithm, but each slice raw to this deep learning algorithm, and had it estimate whether there was EMG activity going on. So blue here is the algorithms estimated based on the deep learning of this image, and green is the EMG activity, or green is the joint angle. And they found that the soleus SO was not as well estimated because it was deeper and not well aligned. But if we look at the gastrocnemius, just based on images, it was able to algorithm, the algorithm in blue was able to know when the joint wasn't moving, or whenever the EMG was not active, whenever there's passive movement, and whenever the joint wasn't moving, whenever there's isometric contraction. So there are kind of possibilities for deep learning that it can pick up features of maybe it's the curvature or some aspect of the shapes of muscle fibers that can estimate EMG activity that's different than joint movement that's passive. And then one thing of almost all these techniques that I showed above, other than that deep learning shear wave technique, which was cross-section, almost all of those are longitudinal, which again, I don't think would really apply to our experiments that we want to do, which is picking up a lot of forearm and extensor compartment muscles at the same time. So there's just a couple of papers actually that have done cross-sectional, especially in the forearm. This is one from Castellini and others in 2012, where they actually, in that case, also just used deep learning. They broke down their ultrasound image into a bunch of regions of interest, shown as bricks in this picture. And then they looked at some aspect of the deep learning, but how much activity there was in a certain region of interest, correlated that with different muscles, and then found the, in this case, they were just estimating position, so not trying to estimate EMG, but found that based on the cross-sectional image, they could estimate the joint position of the thumb and ring finger and other joints. So in cross-section, it is possible to estimate muscle parameters relatively well. This paper is not as important. So I'm proposing, as I was kind of saying, I think this needs to be cross-section. One of the main things is there are just too many muscles in the forearm that I think if we try to do something longitudinally, one, it would just take a lot of different scans, which isn't what we're going for. And two, it'd be too easy to just slip kind of one muscle off to the left or right if we're trying to do this longitudinally. Whereas if it's in cross-section, we can verify what muscle we're in by being able to see the whole cross-section at once. One of the techniques that I'm proposing, we'll see what preliminary data looks like, is that I actually think we have a little bit of an advantage for our experimental setup in the FES, which is that we stimulate a nerve, and then that is like one axial potential volley that all arrives at the muscle within a few milliseconds of each other. That's different than essentially all the other experiments that I presented here, where people are volitionally activating, which kind of ramps up activity. We're just getting a twitch from the patient, and we're trying to figure out, is this twitch correlated with certain muscle activity? So since all we want is those few milliseconds after the electrical stimulation, it's possible that we're able to kind of see some parameters coming either from deep learning or from just some raw parameters that we get that might correlate more closely with EMG. And we don't need to get like raw tension, but we just need that magnitude of initial twitch that comes after the stimulation of the peripheral nerve. So I kind of have 4 ideas that I want to start getting preliminary data on. The first 2 are related, which is just get a cross-sectional raw image like the ones I showed earlier, and I'll show on the next slide as well. And either just create some sort of pretty basic algorithm that looks at magnitude of change, maybe even just pixel change frame to frame, and see how well that correlates with EMG. It's possible it will correlate pretty well, and that could be a really quick method of just getting some non-invasive, really easy to acquire information for all the muscles in the flexor and extensor arms. The second would be to feed it into a deep learning algorithm, which, based on the results of some of the other papers looking through here, could likely be very reliable data for estimating EMG activity of the different muscles in the forearm. I'm interested in the supersonic shear wave techniques. I don't think it applies to how we're acquiring data at this point, which is with single impulses on the nerve cuffs and then getting a twitch out of the muscles. We would have to generate pulse trains, which is doable. It's just not how we're doing it right now, to generate constant tension within the muscles to then see how much EMG activity or how much tension is being generated for that technique to be useful. But once calibrated, it might be really physiologically relevant. And then the last technique, when used by other experimenters, is called TVI, tissue velocity imaging. And it's when you look, and I'll actually just go to you orient the probe obliquely. It's the first step. So instead of being straight up and down, you kind of tilt it a little bit. And then you actually use Doppler to estimate the velocity of whatever tissue is in that frame. So this is just an example of my arm again. We have flexor digitorum sublimis, profundus, and then flexor pollicis longus. Since Doppler is actually just looking at velocity of tissue, you can estimate the velocity of that muscle movement. So after a twitch, you can see that it gives you pretty specific information just to the, or it shows signal that's specific just to the muscle that's being activated. And that's to compare the same image, and I'll pause this here, but now if I'm just twitching my index finger, it will be a different part of the muscle that's moving and it generates a different Doppler signal. So I'm actually kind of going through everything and thinking what might be a good approach that might be giving us physiologic data, which is velocity of muscle movement after we stimulate each contact. All right, that sums it up.
Asaf, are you still there?
Yeah, I'm interested in people's comments, directions going forward.
Hi.
Oh, there you go.
So, Robert, I'm just curious. Can you use ultrasound to measure the activity of orbit muscle? Like, it's very important because nobody has visualized how orbital muscles look like. Different strabismic. Can you look at the orbital muscles in neural causes of strabismus versus orbital causes of strabismus? Just curious.
Yeah, really interesting question. Probably because ultrasounding the eye isn't too hard. You just have the person close their eye and hold it closed, put a little jelly on there. And back inpatient, we had to ultrasound the eye occasionally to look at the optic nerve. I never looked behind the eye, but there's no reason that the ultrasound waves wouldn't travel there well. And yeah, it's possible you can see what the muscles look like, but I haven't looked back there.
Cool. Next time we should.
Yeah.
Any more questions?
I'll just sort of pitch in here. One of the things that I'm very interested in doing is understanding the activation of all the muscles in a let's say in the forearm simultaneously.
Yeah.
As Robert mentioned, we're trying to characterize the effects of stimulation of nerves on muscles in the arm. And right now we're doing that with EMG recordings. And there's a lot of muscles there, as he said, and it would be really nice to be able to find a way to sort of simultaneously gather information on contractions in all the muscles at once, right? So that's one of the issues we've discussed as well. Is that feasible, Robert?
I mean, I think so. I think methods 1 and 4, even in the next few months, if we can ultrasound machine at the same time of these experiments, could immediately be done, which is create an imaging algorithm, look at pixel change right after stimulation of the different regions of interest, and then orient the probe a little bit obliquely and measure the Doppler velocities. And that might be really reliable. I mean, we're collecting EMG data anyways, and we can just put them side by side and see how it looks. And because the EMG, we're not getting all the muscles, we're getting like 5 muscles per time. Once we show that those 5 are correlating well with, if they do, are correlating well with the images of those muscles, I think we can translate that to all the muscles that we don't needle when we see them twitch. I won't put anybody on the spot. I was interested in Bill's comments, but you don't need to turn your mic on.
He took you.
Let's see. Are there comments in the chat? I don't see any other comments in the chat. I'm just curious. So, a lot of times particularly in the eyes, for example, we are very interested in eye velocity, and that is very important marker of various neurological disorders and whatnot, right? And same thing applies to tremor measurements or whatnot, right? So using ultrasound, how reliably you could measure the estimate of velocity?
Quite
Like-
reliably, actually. That wouldn't be a bad technique for it. Maybe showing this image. So the way that Doppler works is typically a square comes down, and then based on how big that square is how slow your frame rate gets. So without Doppler, for example, you can see that this is like 40 to 50 hertz video. You can see every frame really nicely. Once you put Doppler down, it drops it down to like 5 hertz because Dopplers are slow imaging. So it speeds it up a lot to actually just calculate Doppler down a line and not in an entire region of interest like this, which is what we would do once we have the regions of interest that we want. If you were looking at the eye, imagining just a round globe in here, and you did a region of interest velocity vector Doppler on the two sides, and you have a rotating circle, and you have your velocity going towards it on one side and away from on the other, you should be able to get pretty reliable velocities. And those are real, anatomic velocities that would be comparable between subjects, but then should be fundamentally accurate on their own. Just what is the velocity of that eye moving? So you would just have to close one eye, put the probe on it, probably keep the other one open so they can at least fixate with your other eye. I don't think you can do it with both eyes open.
So can you do some correlate of rep stim with this, like you gradually see declining the response in ultrasound with stimulation, like you stimulate and then simultaneously you measure the ultrasound. So not necessarily just for the eyes, but in general, record single units, or instead of recording the electrical activity, which is rather more invasive, you measure the ultrasound as you stimulate the muscle to diagnose myasthenia, for example, like an application. Have you guys been using it? Or, I don't know a lot about this field in standard of care.
We haven't been doing the physiologic types of data acquisition. We've been doing anatomics. So we haven't been doing any stimulation. We haven't been looking at like the magnitude of flexing or things like that. So we haven't been overlapping it with our EMG studies.
But it'll be cool, right? Like how well you see the activation, how fast the activation pattern, the speed of activation pattern, and over time it declines. That means it is fatiguing, right? And you can do that for so for arm muscles, you have access to EMG. For example, for orbital cards, measure like how the whole muscle peters off as there is fatigue that builds up. You see what I mean?
It's probably, yeah. I'll keep it in mind. I'm not sure right now. I think that even with the best ultrasound to EMG correlation experiments, they're getting like 80 to 90% of what the actual EMG signal is. They're able to get 89% accuracy from the ultrasound. Whereas in like fatigue for myasthenia, often it only is like a 20% change. So because we're looking at subtle changes in myasthenia, it's probably, I don't know. I don't want to shoot it down. Maybe there'll be something comes up. But right now, it doesn't seem like something that I would want to switch. I like the very, very accurate EMG that we get from RepStim, all on the same muscle.
Since this is think tank, I'm just kind of bringing up all the weird ideas, but this is another idea, another thought. A big challenge in movement disorders is to diagnose dystonia from
Yeah, so if your connection,
other things, right? So like dystonic muscle
connection
contraction
is really bad, Asif.
dystonic tremor versus like a tremor or whatnot, right? So dystonic is like, can you so the way we like EMG to okay, I'm turning off my video. How about now?
That's better.
Yeah.
Oh, sorry, I'm on my cell phone. That's why. Could you hear me now?
Yes.
You said distinguishing dystonia from, I'm not sure what you said the second aspect.
Like dystonic tremor versus like, so dystonic forms of contractions versus non-dystonic forms of contractions, right? So a lot of times, for example, in Parkinson or in other forms of tremor, you have one component which is dystonia, which is often clinically difficult to differentiate from non-dystonic forms of tremor or oscillation. So just curious. So and we have used a search call system, for example, where we use the kinematics of the position to separate one from the other, and we had been successful in doing so. So just curious whether you could use ultrasound, like position sensing, to answer this question. You can say interesting. We will look into it if you want.
Yeah, I mean, we can talk more about it. You were cutting out a little bit.
Yeah. Yeah. This is just a thought.
I agree. Ultrasound is fast. They walk in the room, you put the gel on, and you have images in a minute.
Hi, Dr. Adams. I just came in towards the end here. Sorry, I'm late. Quick question. My understanding is that you're correlating ultrasound images with EMG during movement, and it looks like it's being pulsed in a nerve stimulation manner, is my understanding.
This one is just me doing this, but the idea would be nerve stimulation manner. Yeah.
Okay, and there's a high correlation between the ultrasound imagery and the EMG. As part of the movement, is the correlation at the onset of movement, at the end, at steady state?
Yeah. Most experiments are at steady state, essentially. So
yes,
are some sort of slow activation, whether it's experiments that are more like, I'm getting lost over here, experiments that are more like slow up and down movements like this they're all kind of steady state. Because if you look, this is like seconds, so it's like 1 second, 10 seconds up, 10 seconds down. So most of it's kind of a steady state contraction that people were looking at for different activations, which will be different than what we're doing, which is more about those little flickers, because we're activating one nerve cuff electrode at a time.
Gotcha. And is it just as accurate for the onset of movement as it is for the fall of the movement?
I don't think I have an answer to that. I don't think somebody's done quite what we're looking at, which is actually, yeah, I haven't seen anybody do electrical stimulation of a nerve to estimated EMG activity. So I guess that was kind of thing I was getting at one point, which is that the typical thing is volitional activity, which is, you're recruiting motor units based on size principle, and it kind of ramps up over a few seconds, which is just fundamentally different than what we're getting with the EMG or with the nerve cuff stimulation, which is that you create a volley of action potentials all at the time they stimulate the nerve that all arrive at the muscle at once. So that hasn't been done. Of what's more accurate, I think it'll just depend on what aspect we're imaging. So if it is something about the change in shape of the muscle or the tension in the muscle, that would need to be at steady state. It's just possible, especially with deep learning, which is hard to say what that's even measuring, that some aspects that changes in that first onset will be the most accurate. I'd say in general, especially for this patient, the offset will not be very helpful, because if there's a twitch and then his arm kind of pronates and then moves, it's like all those extra movements will be less helpful than that initial twitch that we're looking for, is what contact created how much activity in what muscle.
Gotcha.
Other questions for Dr. Adams? Well, we had Asif on there, Robert, we're testing your ability to do signal processing in noisy environments.
Yeah. That's what EMGers do for a living. We're processing muscle sounds. We close our eyes when we really want to hear the signal. Yeah.
Any last comments, Asif? I don't know if you're in a position to do this.
No, I don't have any last comments.
Okay.
Thank you very much, Robert.
Yeah. Thanks, everybody.
Yeah, thanks, Robert.
Yeah. I'll see you all
Everyone have a
around.
good evening.
Yeah, you too.
Bye.